Hello everyone, I hope everyone is safe. Today I am going to present in the topic non-migratory bioactive polymers applied in food packaging. So now let's get started. So these are the topics that will be covered throughout this presentation. Non-migratory polymers are defined to be those for which the bioactive component does not migrate out of the polymer system into the surrounding medium. Typically, this is achieved through covalent attachment of the active component to the polymer backbone. Inherently bioactive polymer backbones or the entrapment of the active component within the polymer matrix. Here in this diagram, this shows the simplified visual comparison of non-migratory and the migratory bioactive packaging. Now we will discuss about the types of NMBP, and types include first inherently bioactive polymers, second polymers containing entrapped bioactive compounds, and third polymers with covalently immobilized bioactive agents. In the case of the first type, inherently bioactive polymers, the structural polymer itself is bioactive in this case. It includes structural polymers with modified backbones. For example, we can take the case of UV irradiated nylon and naturally bioactive materials such as chitosan. And what is this chitosan? It is actually the deacetylated form of chitin, which is a common natural biopolymer extracted from the cells of the crustaceans. And the properties of these chitosan films includes antimicrobial efficacy, mechanical and barrier properties, and they are very significantly affected by this degree of the deacetylation. And this chitosan has been found effective against gram-positive and gram-negative bacteria along with some molds and yeast. In the UV irradiated nylon, the nylon is treated with an excimer laser at UV frequencies 193 nanometer. The induced antimicrobial activity is a chemical change where the amides on the nylon surface are converted to amines. And in the case of polymers containing entrapped bioactive compounds, the large compounds can be immobilized by entrapment in the polymer matrix such that they do not migrate out of the polymer under conditions of use. It requires an active agent larger than the channels present in the molecular structure of the polymer and minimal polymer chain mobility. For example, we can take a polymer with a high glass transition temperature or one that is highly cross-linked. In the case of polymers with immobilized bioactive compounds, an active agent is covalently attached to this polymer backbone. The active agent may be a peptide, a protein, or an enzyme, and it can be synthesized on the surface, or it can be synthesized or extracted separately and then covalently linked to the polymer. Enzymes that are a job on the polymers are termed immobilized. And the developing of this NMBP by immobilization can be explained with two steps. First is polymer functionalization, and second coupling chemistries. Now, after the development of NMBP by immobilization, we will discuss about the applications of these polymers with immobilized bioactive compounds. First, in package processing. This is the most novel form of NMBP packaging. It involves immobilizing an enzyme on the surface of the packaging material so as to perform in the package what would otherwise be a processing step prior to packaging. For example, hydrolysis of naringin, which is one of the bitter compounds in the citrus juices. Here we will immobilize the naringinous enzyme onto the surface of the packaging material. And another example, we can take the case of immobilized lactase, that is beta galactosidase packaging to produce lactose reduced or lactose free milk products at a reduced cost compared to the current methods. This figure is an example of cholesterol reduction of milk with covalently immobilized cholesterol reductase enzyme. Here, the principle of in-package processing with NMBP is clearly shown. Another application is the antimicrobial packaging or shelf life extension. Here, the role of antimicrobial packaging in preventing foodborne illness has two major aspects. First, sterilization or sanitation of packaging materials and prevention of pathogen growth in packaged foods. The immobilization of lactase and glucose oxidase enzymes on nylon pellets has been investigated with the goal of producing hydrogen peroxide to activate the lactoperoxidase system naturally present in milk. Advantages of NMBP. The benefits of NMBP can be divided into four main areas. First, technical benefits. Second, regulatory advantages. Third, marketing aspects. And fourth, the food processor's perspective. In the technical benefits, it includes improved stability of the bioactive substance and the concentration of the bioactive effect at a specific locus. Examples: improves thermal stability of chymotrypsin when conjugated to polyethylene glycol, thermal stability of a small antimicrobial peptide when covalently attached to a PEG grafted polystyrene. Secondly, concentration of the activity at a specific locus within the package and or the food, which this allows the activity to be concentrated where it will be most effective in case of technical benefit. In the regulatory advantages, the regulations revolve around determining if food compounds in packaging materials are food additives. Food additives are defined as substances the intended use of which results or may reasonably be expected to result directly or indirectly either in their becoming a direct component of food or otherwise affecting the characteristics of food. An MBP needs to meet the regulations required of items for food contact use but do not need to meet the more stringent food additive regulations provided lack of migration is proven. Marketing aspects. In case of the marketing aspects, incorporating non-migratory antimicrobials in packaging materials may significantly reduce microbial contamination while providing minimally processed preservative-free food products. This enables to meet the increasing public demand for safe, minimally processed and preservative-free products in the market. The food processor's perspective. A general benefit would be in achieving a more stable product with a longer shelf life, but beyond that, certain NMBP technologies and applications may offer specific benefits. For example, the production of lactose-free milk. Using lactase-active packaging, regular milk could be packed off a normal production run to obtain a lactose-reduced or lactose-free product after a short period of storage. Thus, some of the processing may be accomplished in package instead of in the processing plant, and reduce processing costs and increase flexibility for the food processor. Current limitations of NMBP. The most pertinent limitations of NMBP are limited locus of activity, specific requirements on the mechanism of activity of the active agent, reduced activity, availability of appropriate technology, and an increase in packaging costs. In the limited locus of activity, the need for the reaction constituents to be transported to the packaged product interface is there. This limits the function to areas in intimate contact with the packaging material for solid and viscous liquid foods. And in case of the mechanism of action, here the conformation of the active component in the immobilized state, the location of the covalent link to the polymer, and the mechanism by which the agent interacts with the environment to achieve the desired function must all be considered. One of the limitations of NMBP is its reduced activity. Activity is reduced compared to the native compound. For example, Appendini in 1999 compared the activity of a small antimicrobial peptide when immobilized to PEG grafted polystyrene beads and found that it was 200 to 7,000 times less active than the free soluble peptide. And when it comes to the technological availability, the commercial availability of technology required to produce an NMBP could limit applications. Newer technologies which provide controlled surface functionalization are still in development. And when it comes to cost, NMBP will require further steps and additional materials in film manufacturing or converting processes, thereby increasing the production cost. Intensive modifications such as the attachment of proteins will incur significant cost increases due to the additional processing steps required, the chemicals used in the processing, and from the cost of the agent itself. This will make the production of the NMBP increase its cost. The following points have been listed as conclusion. First, NMBPs have potential applications in intelligent packaging. Second, the use of immobilized enzymes in food packaging is well established and will become more so, leading to in-package enzymatic processing as an extension of the food processing factory. And third, antimicrobial packaging will become another weapon in the war against foodborne disease, and spoilage indicators will help prevent consumption of food that does spoil, leading to fewer food poisoning incidents. And fourth, the technology will increasingly involve the combination of biotechnology and material science, leading to a broader range of materials with a bioactive function. And finally, the understanding of the processes in causing food deterioration will increase, as will knowledge of the structure and function of bioactive polymers, enzymes, and antibodies, leading to still more opportunities for NMBP in food packaging.